All right, so my name is Brandon Stackhouse. I'm from Princeton. I work with the Onstott laboratory there, and I'll be speaking a little bit about this experiment that we've been running for a while, looking at gas fluxes out of these cores we've collected from the high Arctic that have been undergoing this progressive thaw regime, and sort of what we've been seeing there. So we'll be discussing mainly flux and pore gas data, but we'll be touching a little bit on some modeling efforts that we've been doing as well to look at this problem, and a little bit about the microbial community that's present in this area as well. So there's been this sort of amazing body of work that's shown up over the past 10 to 15 years looking at communities in cold environments, all these psychrophiles. And it's gone on this amazing arc. It's gone not just from we see these microbes present in these cold environments where these sub-zero temperatures are frozen stuff, to we see them actually actively metabolizing in these environments, which is incredibly important because if they're just sitting there, you get long-term damage to DNA by radiation or just entropic decay. But if you can metabolize, you can repair your DNA down there. That gives you a much, much longer life to sit in these cold permafrost environments and be a viable organism. Above and beyond that, we've recently started seeing guys that can grow at incredibly low temperatures, 10 degrees C. Recent paper from minus 15 degrees C from McGill here, where we see things that not just metabolize, but they're actually increasing their community size potentially at these very low temperatures. That gives you a chance to just increase your biomass at these areas, not just metabolize, not just repair your DNA. So this has been a really exciting area, especially in terms of its astrobiology implications. And what you see is that these guys are all from permafrost, permafrost, permafrost. So what we want to do is look at these kinds of environments and see how they relate to other areas. So permafrost terrain, for those of you who maybe need a little bit of a refresher, it's this patterned ground that we see in polar regions. So you have the Arctic, you have the Antarctic, and it's dictated by this freeze-thaw cycle that happens every year, where during the summer, everything thaws from the top down to some stable active layer depth where the permafrost begins. And it's this freeze-thaw cycle that generates this very regular behavior in the ground, this very specific type of morphology that happens there. And you develop these large ice wedges in certain areas. You can get ice lenses, so it's this very ice-rich kind of environment in these frozen soils. And it's astrobiologically relevant because much like we see in other areas, it's very low temperatures. So a lot of areas during the winter you get temperatures down minus 40, minus 50 degrees centigrade. And that also means you have a very low liquid water availability for these microbes to exist in, where things are existing either in brines or these very thin water layers on top of ice that's down in the subsurface. So that's a really cool thing that we see these microbes that live in these types of environments. It's also really cool that we see similar environments to this on Mars. So over here we can see patterned terrain from the Utopia Basin, which is up in the northern hemisphere of Mars, right over here, I think. And looking at orbital data, people have looked at regions where they see evidence in the ground features that indicate that there's near-surface stable ground ice in Mars. Not too far below the dirt layer, you'll have solid ice that's present and may extend for a ways down into the ground. And I was agonizing for a while getting this talk ready, trying to figure out how am I going to convince you guys that there's the possibility, even though it's ground ice here, that maybe there's some liquid water here where these microorganisms can exist. And I was so happy yesterday that 2 talks back to back did most of my heavy lifting for me. So the talk that Harvey gave looking at the formation of liquid brines from deliquescence in these salt environments that were seen there, and then the talk by Brendan about these streak slope features that people have seen on Mars that we have analogs for on Earth. And that was fantastic. And so we look at this one, we say, okay, maybe you have these liquid brines, these organisms, the water that's there, it would have to be very salty water that they're existing in. That's okay. The areas that we've done these isolations from on Earth, a lot of them are from these liquid brine environments. They're halophiles. They're okay with that. We look at these streak slope things, and people say if it exists, it's very temporary. That's okay too. These organisms that we see in the Arctic, they're used to only having temporary access to liquid water. During times that they have, the winter, when they're still metabolizing, they get very, very little liquid water. They're frozen most of the time. So salty, temporary, that's okay. The fact that it's there at all, it's very important. It gives them a chance to increase their diffusivity rate, get rid of some of their waste byproducts, get new oxidants in their system. They're happy to even see it at all. So that makes our life a lot easier. So the work that we've been doing is up in Axel Heiberg Island, which we're using as an analog site for some places on Mars. It's not a perfect analog, but you squint your eyes a little bit on a warm day on Mars. Is a nice cold day up in the Arctic. Sure, the pressure's different. You got some oxygen, but this is not bad. This is not bad in the end. So here's our field site. I'm fortunate enough to work up the McGill Arctic Research Station, which is at 79 degrees north. It has a mean annual air temperature of minus 19 degrees centigrade, and it's characterized by being a very low organic carbon soil. We only have about 1% organic carbon that's present in there. And it's also a moist acidic tundra, so our ground is about 4.5 to 5.5 for the pH in terms of the groundwater and the ground soil. So here's a picture of it in the winter. You get this nice snow cover. Here's it during the summer. And what you can tell from this photo is that you really don't have much in terms of plant cover. You're not deriving much of your organic carbon from plants. You have a fair chemolithotrophic community here that's derived most of the carbon cycling that happens in a lot of areas. So we went up there in end winter to drill a bunch of permafrost cores. And if you've never drilled in the Arctic in the winter, it is a special type of hell that is reserved by advisors for grad students they don't think are working hard enough. So I got to spend 2 weeks up there with Guillaume right over there, and we drilled about 41-meter permafrost cores. And it's important that we collected them in the winter because since all the soil was still frozen, it means that the gas composition of the pore water that's there is going to be reflective of what the environment was like when this environment froze at the onset of winter the previous year. So that means as we do our thawing, we can actually go back and sample what that looked like beforehand, plus whatever respiration was occurring during the winter months. So the way our experiment looked is we did this progressive thaw from the top down. Here's just a quick look at the temperature profile where you can see that over the course of several weeks, we progressively brought up sections of the core to above freezing to a maximum temperature of about 4 degrees centigrade. And over the course of this thawing experiment, we were measuring the concentration of trace gases in the headspace, as well as major components. So nitrogen, oxygen, CO2, methane, that sort of thing. And we were also sampling the pore water at several depths down the core. So we had about 5 centimeters, 35 centimeters, 65 centimeters, and then one sampling port that we placed into the permafrost itself. So when we thawed the permafrost, we could get look at what that was doing down there as well. Over the course of this experiment, we also collected soil samples to do DNA extractions to look at the metagenomic profile. Oh my goodness, okay. All right, so we're gonna move on faster here. So looking at the headspace during thaw, we see this release of methane that occurs, and it drops down to near 0 values. We were expecting to see this increase during the initial thaw. It's been seen in other areas, but we didn't see a pickup afterwards, which we were expecting to. We thought methanogenesis would kick in at some point, and you'd see the production of methane going on, which we really didn't see very much. So when we start investigating what's going on in the pore gas during this time period as well, I want you to look at these X's right here. This is our saturated treatments. These are our unsaturated treatments for this thawing experiment. We can see in unsaturated, the pore gas composition of methane decreases rather significantly, and it doesn't in the saturated one. This tells us that in the saturated one, the methane that's down in the subsurface is diffusion limited in reaching the headspace. But if we look at the unsaturated one, we see that it's not. And this should have represented a very large increase in the methane flux during this thaw period. There should have been a large bolus of methane that reached the surface, and we just didn't see it. So we're trying to identify what's actually going on here in this thing. Yeah, so we just have a mass balance issue in terms of where this methane is. And if we look at this data in a slightly different way, here's that same data just shown a little different. If we start running it out and putting methane actually into the headspace, we see that rather than these soils being a source of methane, so we're imagining this would be a large methanogenic community there, we actually see that we have methanotrophy occurring in all cores and all treatments. So mainly just think about this in terms of saturated and everything else. Everything else gets access to some oxygen. Saturated is diffusion limited by the water that's present on the cores. And what we can see is that there's actually, even in saturated cores, methane oxidation is occurring, and we can tell that this is not due to residual oxygen. We can look at oxygen that's present in the pore water and see that this is some sort of non-oxygenic or aerobic methane oxidation, which is very interesting. So I'll skip over this. We did some microcosm work to try to determine what's actually happening in terms of methane production and consumption at various depths and at various temperatures throughout the profile in these cores. And what we used this for was to develop a 2D model for methane production and flux. So this is a coupled biology and physics model that we've made that also uses a pH dependence equation to model methanotrophy, methanogenesis, and it's also linked to temperature by the Arrhenius equation, which is what our microcosms were used to do. Sorry, I skipped over that a little bit. But we're able to make predictions about methane consumption and production in these environments based on empirical evidence that we have. So we've generated our own model for doing this kind of work. So far, we're very good at being able to predict what happens in unsaturated soils. We have a bit of an issue still recreating what our fluxes should look like in terms of the saturated soils. We think this might be due to the fact that we have ANMI going on. I lost a little bit off the side there. There's some interesting papers that show that there's this potential coupling between sulfate reduction and methane oxidation that occurs in certain soils, and we see evidence of sulfate reduction in our soils, which may be indicative of this. So sort of just a quick summary, we've developed a 2D model looking at methane cycle in the Arctic. We see a nice cycle that occurs within the permafrost in the active layer within a linked system, so it's not distal in terms of production and consumption. And we see the production or potential for anaerobic oxidation of methane in saturated soils, and we have some future work incorporating the model with some more things and doing tracer experiments to look at genomic connectivity between methane oxidation and methane production. So, thanks.
Thank you, Brandon. Any questions?
I've got one online if you want to take that in the meantime.
Sure.
If ice microbes remain viable for thousands of years, how long can microbes remain viable in permafrost environments?
For a lot of these guys, as long as they're able to, as long as the system isn't completely closed, you get some cracks in the system, they're able to get fresh oxidants, and they can turn over their DNA and do some DNA repair, there's not a good reason that they can't exist for thousands and thousands of years in the permafrost. They just need to be able to have a minimum energetic requirement met in order to do that base repair. So they can last quite a long time down there.
Do you have any 16S data yet for what's going on in those communities, or still waiting on it?
I'm sorry, could you repeat that?
Do you have any 16S rRNA gene profiles for the communities in these cores? Are you still waiting on that data?
Yes, so they're about 1% archaea in terms of what's going on. So there's a fairly low, presumably methanogenic community that's going on there, and they're primarily focused in about 65 centimeters or below. We've gotten some stuff back looking at the top 35 centimeters. We see about 1% alpha proteobacteria that should be methanotrophic communities, type 2 methanococcus, or methylococcus. We're still doing work and looking at what the change is over time. We'd like to see a difference in the profiles as thaw goes on, but we're still waiting on getting that data back.
Any other questions?
Do you plan to screen any of the permafrost isolates at Martian conditions? And I ask that because my lab recently got a isolate from permafrost in Russia that grows at Martian conditions, so you might have something exciting in your permafrost.
That would be awesome. I'd love to do it. If you have any money for me too, I'll be taking checks, cash.
Hey, so essentially you just showed that you have a methane sink in thawing permafrost.
Yes.
And so I know this is an astrobiology conference, but would you care to comment on the inevitable conclusions that people will make that with global warming and your permafrost laws that this might actually solve all of our problems?
Yeah, so this actually presents a really unique environment to look at this kind of thing, where it's a low organic carbon soil, which is very different than a lot of the research that's done in terms of methane release into the environment. Those are usually done in very high organic carbon content, like peat bogs, that kind of stuff where you see 20, 30, 40% organic carbon. We have a really hard time here actually turning on our methanogenic community. We feed them acetate, we feed them H2CO2. They don't seem to really like it, and our methanotrophs are super happy to eat any methane that we do give them. So this might be indicative of types of soils, low organic carbon, a little bit drier, where we've just sort of crossed some sort of threshold and they turn into sinks as opposed to sources. I think it depends on what the distribution is, but areas like this look like they may be an offset to methane increase in Arctic fluxes.